So in this talk, I am going to talk about two case studies that will illustrate the types of stress that wild animals cope with in nature. The first example is going to be a relatively unusual one of the potential stresses that may have induced the evolution of amphibious behavior in fish. The second example is in the case of eastern grey kangaroos and their response to stress resulting from environmental change, in particular changes in rainfall, for example, drought. But let's start off with our case of amphibious behavior in fish. So many of you will automatically think of the iconic mud skipper in the context of amphibious behavior in fish. So the reality is, though, that mud skippers are actually mildly amphibious. They're essentially scubaing on land. They're holding pouches of seawater in their gill pouches, and they're essentially respiring based on that, and then returning to burrows in their mangroves, which are full of water. So they're actually still heavily tied to their aquatic environment. But if we step back a bit and look at the evolutionary tree for all living fish, which is what's on the screen here, and we go into the scientific literature and look at all the different types of examples of fish coming out of water, we can see that it's actually relatively widely distributed across living fish. To such an extent, there appears to be at least 130 different examples of the invasion of land from fish, from minutes to several days to essentially permanently, which is what we're going to talk about just now. So a less well-known group of amphibious fish. So you all know the mud skippers, but you probably don't know about these intertidal blennies that I've got on the bottom of the screen here. Now, this is a really remarkable family of fish. We've created an evolutionary tree for all the different species within this family, and we've reconstructed the behavior of these fish. So we just observe them in the wild, and we use various statistical methods to then map on the evolutionary history of the change in that behavior onto that evolutionary tree. And the take-home message is that within this single family, it appears that the independent repeated evolution of amphibious behavior has occurred at least 7 times. And this is really interesting because it means that there are examples of different species at the different stages of the transition from an aquatic environment out onto the land. So we've got obviously the aquatic ancestors, but we've also got species that are exhibiting varying levels of amphibious behavior. And we even have species that are exclusively terrestrial that never return back into the water. And I'll show you a video of those species in just a moment. So in terms of within the blennies, the repeated invasion of land is a global phenomena. It occurs on multiple islands in the North Pacific and South Pacific, and even into the Indian Ocean as well. We've gone to various islands to study these amphibious fish, but I'm going to focus on those blenny community that's on the island of Guam, which is where we've done most of our work, because there's quite a diverse range of different blennies on this island. So Guam is essentially a rock in the middle of nowhere. I suppose the closest landmass is probably Taiwan or perhaps Japan. So on the island, this video is showing this terrestrial species. This is the species that does not return back into the water. You can see that they're spending quite a bit of time grazing for algae and detritus on those rocks. They seem to be really agile. They seem perfectly comfortable moving, crawling around, and even hopping from one rock to the other. The thing I want to emphasize also is the fact that every time there's an incoming wave, they are actively trying to avoid it. And this is because they've adapted their morphology to such an extent for being able to move about on land that they're really probably poor swimmers as a consequence. You might be asking yourself, how on earth does a fish breathe out of the water? They still use their gills, and they also breathe through their skin to some extent as well. You would have noticed from the video that it's very wet, and they have to stay within that splash zone of the intertidal zone, because if they dry out, their gills collapse, their skin starts to fail to respire, and they essentially suffocate. But as you saw from the video, they're very agile on land. They seem to be perfectly comfortable within that splash zone. So that's the terrestrial species. But again, we have multiple separate indications of different species that have got amphibious behavior. So they're spending part of their time in the water, part of the time on land. So you might also be wondering about, well, why would a fish come out of the water? I mean, surely, there has to be a reason. So that is a very general question in science as well that is applicable to any organism that moves from one environment where it's presumably well adapted into a completely different environment where presumably it's less adapted. So why is this change actually occurring? It usually boils down to two reasons. Either that organism's trying to escape something in their ancestral environment, or they're trying to find something new that's available in the different environment that's not available in their ancestral environment. And this is equally applicable to the evolution of amphibious behavior in fish, and in particular these blennies. We're going to focus on that first order scenario of escaping something. So what initially prompts a fish to move out of the water onto land? There is a very old idea, a hypothesis that essentially what's occurring is that fish are voluntarily stranding out of the water under a rock or in rock crevices, and waiting for the incoming tide because it's better in that terrestrial environment than it is in the rock pools, because the conditions in those rock pools at low tide are really stressful. So the idea here is this is a photograph of the intertidal zone on Guam at low tide. And you can see if you're an aquatic blenny, you'd be limited to residing in these rock pools until the incoming tide. And so the idea is that you're in the tropics, the water heats up, it becomes too hot, so you're potentially exposed to heat stress. But in the process of heating up, the water is also leaching oxygen, so there's less oxygen in the water for blennies to respire effectively. There's also this notion that evaporation will reduce the rock pool and, as a consequence, increase the salinity of the water as well. And salinity is actually quite a bit of a physiological challenge for fish as well, even for marine fish. So I've got an animation here that basically summarizes this very old school hypothesis about why fish are coming out of the water. The water heats up in the sun, leaches oxygen, evaporates, increasing the salinity. So it's a very intuitive idea, right? It seems to make sense to such an extent that people really haven't explored to the extent that this is actually the reason why fish are voluntarily stranding out of the water to escape these abiotic conditions. So we've actually done a study last year where we measured the conditions in these rock pools at low tide. So all these rock pools that become isolated on Guam are highlighted here by the white circles, and we measured temperature and oxygen and salinity in these rock pools. And I've got a photograph here of a typical rock pool isolated at low tide, and you can see that I've also highlighted there that there are a bunch of fish in that rock pool. Interestingly, those fish are the amphibious blennies. These are the blennies that are capable of being in the water, as you can see, and spending time out of the water being amphibious. And yet, at low tide, they are choosing to stay within that rock pool. So even on that anecdotal observation, that old school hypothesis is a bit shaky. But let's have a look at the data. So if you're not familiar with looking at data plots, this is what we call a histogram. Each one of those bars represents the number of rock pools that fit within that particular environmental range. And the bars are color-coded based on dark bars are rock pools that have aquatic and amphibious blennies in them. And the white bars are ones where there are blennies completely absent. And you'll see straight away that there is a wide variation of environmental conditions within these rock pools, but blennies are across the board. They are quite tolerant of really extreme changes in temperature. Those rock pools do heat up quite dramatically in the tropical sun, to such extent that our instruments maxed out at 36.5 because we weren't anticipating it getting hotter than that. But you can see that, there are blennies in those rock pools, despite it being really, really hot. There are blennies in rock pools with very low oxygen. The vast majority of the rock pools actually have higher oxygen at low tide than they do when they're swamped with seawater at high tide. And this is because you've got things like algae generating oxygen through photosynthesis and basically pumping oxygen into that water. So that doesn't seem to be a big deal. Salinity, rather than increasing, it actually is lower in many of these rock pools. And this is because there is rainfall runoff that accumulates higher up on the landscape and it progressively trickles down into the water. And at low tide, it goes into these rock pools and changes the salinity down into a more brackish, low salinity level. And yet, these blennies still seem to be relatively tolerant, even with those wide changes in salinity as well. So these rock pools are actually stressful for most fish, not these blennies. So in terms of prompting these fish to come out of the water to voluntarily strand, to wait for the incoming tide, this is not the reason why amphibious behavior has evolved in this group. So what has prompted this group of fish to actually start coming out of the water? Is it something else? Now, another stressor in the environment is a biotic one, and it's predation. Now, predation is ubiquitous in nature. And in the intertidal zone, it's particularly bad. The intertidal zone is a major area for fish predators to be hunting their prey. In particular, small blenny prey are particularly vulnerable to aquatic predation. So it's a stressor. So perhaps this is something that they're actually trying to escape from the water and coming out on land. There's two of my graduate students, Tom Summers and Courtney Morgans. They used blenny mimics. Now, this is essentially creating plasticine models of blennies. It sounds really strange, but the idea is you put this out in the environment and predators are fooled by them and they try and, attack them and they'll leave their impressions or their bite marks in that plasticine and we can see whether which proportion of those mimics have been attacked compared to those that haven't in the different environments. Now, it's plasticine, it's food grade. This is the sort of stuff that people give their kids to play with. So even if those predators are actually munched on that plasticine, it's not going to do them much harm at all. So they went out there and distributed this out and they found that the probability of being eaten, so being attacked, and we've got some examples on the screen here, is really high in the water. There's roughly a 50-50 chance of being predated upon over about a week staying in the water in the intertidal zone. In comparison, the same time period on land, it's less than 10%. So there's still predators out on land, still attacking these blennies, but vastly fewer than if you're actually in that really nasty intertidal zone. We've also done transects where we've actually mapped the distribution of amphibious blennies and their predators. And it turns out that at low tide, yep, they're quite happy to stay in those rock pools because there's very few predators in there to begin with as well because it's too small. With the incoming tide, however, brings all those fish predators as well. And what is happening is those amphibious fish are choosing to come out of the water as the tide comes in to escape those predators. So it's a very different scenario to what we actually thought was going on. So life is actually better out of the water, particularly at high tide, where there's all those nasty predators underneath the water. And we believe this is probably one of the original reasons why these fish originally started to come out of the water when they were still aquatic to try and escape predation. So there is an adaptive benefit to it. So stress in the wild is ubiquitous, it's everywhere, certainly in the case of predation. It's also species specific. So those conditions in the rock pools would actually be quite a struggle or a challenge for many tropical fish, but not these blennies who seem to be reasonably tolerant to really extreme changes in temperature, oxygen, and salinity. So in terms of the three R's, this is replacement, reduction, and refinement. This is essentially the code that we use in science, in animal research, to maintain animal welfare and ethics. I will just point out that we're actually not catching any animals here, and we're using replacement. We're replacing them with, blenny plasticine mimics to measure predation. And yet we can still, address some really big evolutionary questions with virtually little impact on the animals themselves. So it's a really wonderful strategy to take. So the second case study that I'm going to talk about is the eastern grey kangaroos in the New South Wales Central Tablelands. This is at the Woollahra long-term monitoring site. It essentially started out as a mental health break for me from the admin of academia. I thought, what can I do for a few days that will just, let me decompress? And I thought sitting in a hive with my camera and taking pictures of kangaroos would be really great stress relief. And it really is. I've been doing it every single year, September, October, every single year, but go out there for about 4 or 5 days to take pictures of kangaroos, just like you got on the screen here. Quite reasonably, you might be asking yourself, what on earth would you do to that? I mean, you can't do science with just photographs, can you? We can. We've been using a program that's been designed to trace the shape of the fluke of humpback whales. So the tail shape is specific to individuals. We've used it to manually, map the inner ear of these kangaroos, which is specific to individuals. It's essentially a thumbprint. And we can actually determine who is who from one photograph to the next. Now, it takes a huge amount of time, and it certainly wasn't a mental health break when I got back to campus with all these images. So I actually stopped doing this because it was just too much work. And those photographs accumulated and accumulated, and we had thousands of these things until Nora Campbell came along in 2020. She was looking to do an honours project during all the lockdowns. I told her about this library and she was really keen to actually go in there and manually do all this sort of stuff to track individuals from one year to the next. And this is what she came up with. It looks really complicated, and it's like, what on earth is going here? Basically, each line is an individual kangaroo. She found over 150 different individuals in this population, and she was able to track them from one year to the next. To illustrate, this is female number 4. She was first identified as a subadult back in 2015, and she has been identified in every single year since. You also might be able to see on the screen here that you can see from the photographs when she's actually got pouch on. So we can actually get information about when this particular female is reproducing and in what years. So what else can you do with all this information? It's like, what can you get out of it? So Nora actually developed these social networks because you can see which individuals are associating with each other because they're standing next to each other in the photographs. And so she used that to be able to form these network graphs to be able to determine whether the social connectivity, who's associating with who, is dependent on the sex of the animal, it's dependent on the age of the animal, whether the animal has young or not, no young. And one of the key things that Nora discovered was, in particular for adult females, these females are often hanging out with the same other female or group of females from one year to the next. So they're forming these long-term social bonds and they're hanging out together over years. And this is something that we really didn't realize for any of these macropods, so kangaroos, wallabies, and wallaroos, and certainly not for eastern grey kangaroos. So it was a remarkable discovery, just basically pictures taken from a hide. The other thing we're exploring at the moment is this is an example of a male who was spotted as a subadult in 2016, and then again in 2018. You can see he's grown up into a very large mature male. And that gave us the idea that perhaps we can actually measure individual growth rate. So we're still exploring this idea, but there's a lot of potential here that we're going to try and leverage in future studies. So this is Nora's data, but she also just counted the number of animals from one year to the next. It's color coded depending on whether it's a male or a female or pouch young or subadult. And all I want to show here is even with these gross estimates of population size, you can see that the population fluctuates quite dramatically from one year to the next. So because Nora has tracked these individual animals, we can actually determine who's disappearing from the, permanently from the population and what is it reflecting. Now, we've gone through one of the worst droughts on record that really impacted this particular field site as well. This drought was several years in the making. It reached its peak and ultimately broke at the end of 2019. Actually, it broke at February in 2020. And from that point on, it rained almost continuously for 3 years after that. So it's a, large oscillation in terms of rainfall. In general, the populations, regardless of the environmental changes, females that don't reproduce are more likely to disappear from this population. Males are also more likely to disappear from this population as well. In bad rainfall years, so in severe drought, but interestingly also in the worst flooding on record as well, um, everyone is equally impacted. You've got a 50-50 chance of disappearing from this population through mortality in bad rainfall years. Not just drought, but high rainfall years as well, which was unusual. And they started to think, is there a way we could use these photographs to get a visual health check of these animals? So some zoos use this kind of Gauntness scale, which I have up on the screen here. It's very reminiscent of the Gauntness scale that's used for lab mice and rats. But in terms of, macropods, say kangaroos and wallabies and wallaroos and things, it's pretty limited because in the wild, you're not going to get overweight animals. It just doesn't happen. And you're not going to get severely underweight animals either because they're going to be taken out by predators way before they even get to that stage, which also probably rules out the underweight animals that they've got on the screen here, where you can actually start to see their ribs and things like that. You generally don't see that in wild populations. So we thought we could use a more quantitative method where we just kind of measure the width of the forearms, so the muscle and the fat stored there relative to the very bony wrist that doesn't change on condition because it's the skeleton. Good years, it's fat up here relative to this, and bad years, it shrinks in that individual relative to the rest. So that's the idea behind it. And so we've recently got a grant from the UNSW's 3Rs grant scheme to try and explore this condition score and a bunch of other ones. The hip to waist ratio, it's very reminiscent of what we have in humans. In macropods, they store fat in their tail. So we're trying to figure out whether we can use the width of the tail relative to body size as another condition score. And we're in the process of benchmarking this data to captive populations in tourist parks, for example, where tourists are constantly feeding these kangaroos. So they've also got veterinary care 24/7. So they're in ideal condition. And we're using that comparison as a benchmark for these wild populations, just to see to what extent we can actually use a condition score based purely on this remote, ratio index from photographs. Now, I mentioned that we're using this package to trace the inner ear. It's a huge amount of work, and thanks to Nora, we've got lots of data on it. But if we can't identify those individuals, all the stuff that I've just been talking about just goes out the window. So you need to have individual recognition here. This grant we're also using to work with computer scientists to create a new software package that's reliant on AI that we can feed in these photographs without any processing, and it will tell us who is who. And we're going to package this together and make it freely available to everyone, to the public, to in particular wildlife managers as well. So our hope is that with a combination of these condition scores and this software, people can now start to monitor the health of these wild populations without having to catch these animals. So measuring the impact of stress in the wild is actually quite hard. We had to come up with these condition scores and, tracking animals from one year to the next. The impact of stress from environmental change is individual specific. Some individuals are going to be more vulnerable than others. And also we discovered some unexpected causes of stress. Sure, drought is bad. That seems to be obvious. What we hadn't anticipated is that high rainfall. We actually thought that, rainfall, lots of vegetation on the ground, lots of food, lots of feed, life should be good for these kangaroos. But it turns out that in extreme high rainfall years is just as bad as in extreme rainfall years as well. In terms of these 3Rs of the ethics of scientific research, we're replacing the use of animals here. We're not catching them to do these health checks. We're trying to do this all remotely, and we're refining these index of condition and, being able to individually identify kangaroos without catching them, putting tags on them and all that sort of stuff. So we can address lots of really interesting big ecological questions without actually catching these animals and interfering with these animals at all. So again, another wonderful win-win scenario. Now, if you're interested in animal behavior of wild animals, I've created several video stories, 8 episodes up on YouTube. We've got the QR code up on the screen here. Just scan that and it'll take you to the landing page. It's deliberately designed to be accessible to lay people as well, so you don't have to be a scientist to follow what's going on in these videos at all. I'd love to hear from you. Send me an email or check out our research page for the more kind of academic-oriented scientific research.